Great. Welcome to the ALS-ENABLE webinar. I'd like to introduce Paul Adams, who is the PI for this P30 grant at the ALS, and he'd like to say a few words to welcome. Paul, that's all you.
Great. Thanks, Jay. Hi, everyone. On the webinar, we can't see you, but we can see there's a bunch of people signed in, so that's fantastic. Thanks for spending the time to join us today. We were hoping today to give you a chance to hear about what capabilities we have at the Advanced Light Source here in Berkeley. And that's especially relevant because of the imminent shutdown for about a year of the Advanced Photon Source for their upgrade, which is an exciting time for them and for the US to have an upgraded synchrotron. And we're looking to our upgrade at Berkeley a couple of years from now. But we want to make sure that the community knows what is out there to help with collecting your data and doing your experiments during this time when there will be distinctly fewer beamlines available. Advanced Photon Source, of course, has a lot of structural biology beamlines, and they will be unavailable for about a year. So today we're going to have a number of presentations. So you're getting the information, including how to access the Advanced Light Source, to be able to sign up, apply for time, and use the facility. And we also, of course, will leave time for your questions. So we hope to answer any questions you may have. So I unfortunately have a bunch of other things I have to do today, so I won't be able to stick around for very long. But I did want to say hi to start things off, and then I wanted to pass things back on to Jay, who's going to give a brief overview of the resources, and then we'll get into a few details from others. So take it away, Jay.
Thank you, everybody. I'm Jay Nix. I am the Beamline Director for the Molecular Biology Consortium, but also a happy member of the ALS-ENABLE team. Today, instead of going through each beamline and all of its capabilities, we thought we'd just give you an overview of all of the beamlines and the capabilities, because frankly, they are very similar. But I asked our beamline scientists and leads for each of our technology centers to give a little taste of what is offered beyond the normal that we do, and hopefully inspire you to use more than just standard MX. But most importantly, we're here to show you how to get time. And Andy Jones from our user's office will help walk you through that. So ALS-ENABLE is a mature synchrotron research grant from the P30. We use this money to support our general users, improve our beamlines, but not only general users to MX, but we also offer biosacks and footprinting, which Greg Hura and Corey Ralston will be speaking about later. Our beamlines are different in many respects, but they all do much the same thing. Some of the differences, though, are some of the beamlines that Mark will be talking about, Mark Lair from the BCSD, actually offer fully automated data collection. There are collaborative crystallography projects spread between a number of the sectors. James Holton has a strong room temperature data collection program. He can also, at 831, actually do plate data collections and plate screening, which I don't believe is offered anywhere else in the MX at the ALS. We'll also be joined by Bonnie Shankaran, who will tell us a little bit about the collaborative crystallography projects. And I'd like to move on now, just to mention that on the left-hand graph, basically this is the number of publications from 2017 to 2020. These are raw numbers. Nothing's been put towards like per beamline or anything like that. But what you can see is that big orange graph that dominates, or big orange line that dominates the graph, that's the APS. And that's all going away next week, which kind of explains why we're here right now and how to help you guys get your data collected as we go into this dark period. On the right, you can see how the ALS stacks up as far as, the high profile papers per resource. And that's something we always like to bring up because the ALS was built as a low energy light source, but by the addition of wigglers, undulators, and superbends, we've been very productive in the high energy range. And so what we'll be talking about today is, of course, macromolecular crystallography to get our atomic resolution structures. We have 7 beamlines. It's funded by industry, HHMI, the NIH, and various universities. Small angle scattering at the Sibbles beamline 12311, Greg Hurra will be telling us about, and how we can actually look at biomolecules in solution and how we can combine that with crystallography and soft X-ray tomography. And I have 10. And Corey will join us tell us about X-ray footprinting, which is just ramping up and is very exciting. And I'll tell you a little bit about our participating research teams. The PRTs, this is how the individual beamlines are funded at the ALS. And we're made up of academic and industry partners. And they have guaranteed beam time of 75%, and we offer 25% to general users. The BCSB and HHMI in sectors 2, 5, and 8, the MBC in 422, and Tom Albertron with UCSF at 831. So how do we get access? General users can go through the general user proposals that Andy will walk us through. It's administered by the advanced light source, and you can either make proposals for 6 months or put in rapid proposals, which will generally be given time within about a month's time, sometimes faster. One of the things that's a good idea, that's a refrain from this talk, will be contact a beamline scientist. We'll help you get through the things and get you through as expeditiously as possible. These general user proposals are merit-based and peer-reviewed. So that also adds a little bit more time to it. For those proprietary research users that would like to use the ALS, this requires a proprietary agreement with the ALS first, and it does incur some costs for reimbursement of the photons to the DOE. The general user proposals, absolutely free. And once again, for those that would like to bypass the whole peer review process, the PRTs have guaranteed time to our users, and we can assign that time as we see fit without having to go through the peer review process. Here's the contact information for each of the PRTs or the individual beamlines. Just wanted to have a picture of this since we're recording, and these slides will be available soon on our website. So just a general overview of the MX beamlines at the advanced light source. There's 7 of them. The latest one Mark will be talking about, 201, is from an undulator source. Sector 5 is from wigglers, and sectors 4, 8, and 8 are off superbands. I'm not going to go into flux. I think James has very good things to talk about that, but generally they're in the 5 to 10 to 11 to 5 to 10 to the 12th range. Plenty of photons to get what you need. We have different ways of focusing our beam size at the various beamlines. In 821, we use fixed size apertures at 30, 50, and 100 microns. The BCSB beamlines also have constantly variable collimators where you could dial it from 10 to 20 microns up to 150 to 200 microns. We could do the same thing at 422. I'll talk about pucks and robotics coming up, and sample capacity you can see there. The main thing I wanted to say though is that all of the beamlines at the ALS are now upgraded to some type of shuttleless data collection technology. Most of them are made by Dectris. We have the Pilatus 6Ms, 2Ms, the Iger 2 9M. 422 has a CMOS-based shuttleless detector. The frame rates that you see on there are for the detectors. Talk to the beamline scientist about what's an appropriate frame rate to use at the individual beamline. Like I said, we have robotics in all of the beamlines, both for on-site or remote use, also for collaborative crystallography. All of the beamlines can handle the Unipucks or ALS pucks, and we strongly recommend using these pucks because they're the easiest to get loaded at all of our beamlines. It also makes access easier since if you're putting in a rapid proposal, we will try and find the most appropriate beamline for you to use. And part of that depends on what pucks you're sending your samples in. 422, 821, and 822, in addition to the ALS and Unipucks, can also use the Rigaku actor pucks. Additionally, 831 can use SSR cassettes as well as ALS pucks and Unipucks. James wanted me to include that we can also use, you can also send us samples via vials, even with plastic caps, we can get them up there. I put 422 on there reluctantly because I don't want to encourage this too much, but I will say if you send it to me, I will get them loaded, but I will not do plastic caps. We'll have to use the standard metal bases. But if you want to bring them on site, go ahead, knock yourself out. The beamlines can also handle all of these standard bases that you could get from a Hampton or Mighty Jet. I will say this, spine pins cause the most problems for the different robots because it is a slightly different size. And the shoulders on, the ones with the shoulders can cause problems in, well, I know in Sector 8 and Sector 4, we've had problems. And so, yeah, that's a general overview of what we'll be talking about today. And next up, we'll be talking about technology developments and what's going on within the ALS-ENABLE team. And I'm going to introduce Mark Allaire of the BCSB.
Thanks a lot, Jay. Thanks. Great introduction to all the beamlines. So I'm going to speak on behalf of the Berkeley Center for Structural Biology and a little bit focusing on the latest toy. Latest beamline on the floor, and as well talking about automation. So Berkeley Center for Structural Biology is supporting a 6 beamline on 3 different sectors. And as Jay mentioned, if any of you guys are HHMI, then you could get direct access to our beamlines because we're supporting the HHMI facility for principal investigators. So, it's a big team behind. I think we have a lot of people on this webinar, but there's a lot of people behind that keeps the beamline operation. And I just want to be sure that you see their face when you will start using our beamlines. So we were talking about the latest beamline on the floor, Gemini, beamline 201. Basically, that's first micro focus MX beamline at the ALS. Basically use monochromator, silicon 111, and or as a multi-layer as well. So, and the focus size can be as small as 30 by 30 micron. Indeed, we've been achieving better than 20 by 20 micron this time. So, it's a real, I think we have this undulator-based X-ray source, which is named LIDA. But LIDA is in the ring right now, and we've been achieving a first light a couple of years ago. And more importantly is that the beamline is now fully equipped with all the optics and basically the end station. If we have this double crystal monochromator, silicon and multi-layer, like I mentioned. There's a pair of KB mirrors that fully focus the beam down to the spot size that I was mentioning. We have certainly some slits diagnostic, and in the end station, we have this Pilatus 36M detector, a fast one. And as well the smart gonimeter and the NAT X-ray robotic system, which can include the 24 box. The beauty of Gemini is that the science is starting with the small spot size. We just got our first PDB deposit last December, and we're quite happy to continue with the success and the success of your project that will be sent to the ALS-ENABLE beamline. So, for the BCSB beamline specifically, this is the interface that you are going to use, the B4 graphical user interface. In order to do that, you'll need the BCSB user account, and then All the connection to the beamline are done through an X client, remote access. We've been told that this interface is quite user-friendly, quite self-intuitive as well. And there's tons of automated tools that are available, and we could certainly provide another video later about what could be done. But the ultimate tools that we have is that basically you have a fully automated data collection pipeline, like Jay was mentioning. So you can ship tons of viewers, tons of samples, and then we could pull that to the pipeline, and then it's about 6 minutes per sample. And then if you're going to get the data sets with all the data sets processed. So the approach in this automated pipeline, it's doing loop centering. And then after that, on the loop, it's going to do a raster approach in order to find indexable spots in the rastering. And then from that indexable spots, it aligns center 90 degrees away from that. And basically, you would get your full data set. So this could be used for screening for data sets. And right now in Gemini, we're using the automated pipeline to get all the data set that we need. So that you will need as well. So thanks a lot. And at this point, Jay.
Thank you, Mark. Some fun stuff happening over there. Next, I'd like to introduce James Holton, director of the Tom Albertron and Beamline 831. He's going to give us some thoughts on radiation damage.
Yes, thanks everybody. Hi, I'm James Holton. I run Beamline 831. And one thing I just realized we should tell you about is this quick excitation scan / fluorescence scan experiment. The beamlines that have vortex detectors can do this. The nice thing about ALS-ENABLE is you don't need to worry about that. Just tell us the science you want to do, and we'll assign you to a beamline that can do this. This is a 10-second experiment that'll tell you all the elements that are in your sample. So if you got some site, could be a zinc, could be a nickel, you're not really sure, this will resolve that. It's very fast. Doesn't even do a lot of radiation damage because the beam is heavily attenuated for these scans. And also, while I'm at it, this is a picture from, you might recognize this from Blue Ice. At 831, we actually run native Blue Ice. We co-developed this, collaborate with this with Stanford, and we try to make it as similar to that interface as possible. All GUIs operate more or less the same way, but this, if you're used to Stanford, this would be, or other Blue Ices around the world, this will be an easy adaptation. And we do support their unattended data collection via the screening tab. And as Jay mentioned, we support a lot of different pins. We don't do those because they're weird, but anything else in this picture we actually can support. There's only one beamline that does this, so you have better luck getting time with standard pucks. But if you're poor and you only have these old kinds of pins, we can still help you out. We can also shoot through trays. This is something that is still under development. I believe you can do this at Diamond, and there's a way you can do it at Stanford now too, which I'm helping out with. But essentially, this is a way of shooting your crystal without ever having to touch it, which can give you useful information. But I also want to tell you about some radiation damage stuff. Now, radiation damage is really important to understand because as all of you are going to be moving from one beamline to another, the relationship between exposure time and damage varies by a factor of 10,000 depending on which source you're using. So you want to know what you're up against. Now, this movie is what happens when you turn off the cryostream after shooting a crystal. The neat thing about it is this little guy over in the corner, that actually survived. That still diffracts after the big crystal explodes. And that's because the beams here at ALS are very well collimated. So the quick thing you need to know about damage is that it fundamentally limits the amount of data you're going to get from any given sample. And it actually doesn't vary much from beamline to beamline. Caveat, of course, for active sites. If you have interesting chemistry going on in your active sites, you need to worry about radiation damage. It unfortunately can't be outrun. There have been many attempts to do this. X-Fells can do it a lot, but they have a different kind of damage. So in general, damage cannot be prevented. The good news is it can be corrected if you collect your data the right way. And it can also be predicted. We've been studying this for 20 years and we understand how to calculate what the dose is going to be. So this is the QR codes to a paper that describes this, but these are the most common kind of mistake people make when they have especially sensitive crystals is they actually accidentally put in a heavy atom. Iodine being a common one where you, let's say you put 4 molar sodium iodide, doubles every half molar, so that means your crystals aren't going to live very long. And so this is something really good to bear in mind, sending your crystals to any synchrotron. Also, I have a hobby of trying to accumulate records of all the studies that have been done on radiation damage and the dose limits of various types of chemistries. They're not all tabulated, but if you have something similar to one of these, then you can help predict. Again, that QR code will take you to those studies. At this point, you might be wondering, what's this megadray thing? It turns out that's the international standard for dose, which is in joules per kilogram. And we have assembled a document, which is linked to that QR code there, which has listed the flux of every beamline in the world, including the one you're used to using. And it will also tell you how long crystals will live and how long your chemistry will take to transform your active site. We've also distilled this down into a couple of web calculators. This is being slow to update here. There we go. This calculator will tell you how many images you will get from your crystal. There's a pull down on top, which has every beamline in the world and their standard fluxes. And the second calculator, it will tell you whether or not your experiment will succeed because of radiation damage limits. You put in things like what resolution you see on a snapshot and how big your crystals are, and it'll tell you whether or not you need more than one crystal to solve it. Those are useful tools. And we incorporate these technologies into all the data collection software here at ALS. But the critical thing I wanted to tell you about in my last minute is that the number of photons you get from your sample is independent of which beamline you use. You get about 1 million photons per cubic micron under cryo. And that is a fixed number. So all you get to decide is how many images you put those photons on. And when you're making that decision, you kind of want to do as many images as possible to optimize correction for radiation damage. And it turns out that this image and the one that follows, if it ever shows up.
It's showing up on Zoom before the screen.
Yeah, it's showing up here before that. Anyway, so the long exposure era has come to a close. Because it turns out we've done these experiments, and if you collect data really quickly with lots and lots of images with the same number of photons, you actually get the same resolution if you process it with the right software. And this is actually how we solved the CRISPR-Cas13 structure with Jennifer Doudna's group. So this does work, and the optimum you want to shoot for is 1 photon per pixel. And that gives you the maximum capability to correct for radiation damage. And I will close up here. Know your dose, avoid those world records, keep your atoms spinning, and try to keep your atoms light. And who's next?
Thank you, James. Next, I would like to introduce Banu Sakaran, who's going to tell us about the collaborative crystallography projects.
Okay. Hi, good afternoon, everyone. I'm Banu Sakaran. Will be giving a brief presentation on the CC program. So collaborative crystallography is a peer-reviewed service crystallography program, and it's only for the non-proprietary users, and it started in 2008 at ALS. And we helped users determine close to 450 structures published in over 150 peer-reviewed manuscripts. Primarily, we help provide scientific services in the form of data collection and data processing. Depending on the need, we further provide support in structure solution, refinement, and validation. And we also help in depositing structure and data deposition as well. And the beamtime proposal management, we also help in submitting the proposals. And also during the manuscript, we also help in writing the manuscript. And we also train the students and postdocs and research assistants by giving demo or training them in data collection and data processing, and also during the refinement and modeling their structures. So how does it work? Like, you basically submit the rapid or a general user proposal to ALS, and you have to choose the ALS-ENABLE box, and underneath you have the collaborative crystallography box. You have to click on it while you're submitting your proposal. When the beamtime is awarded, as I mentioned earlier, it's peer-reviewed. So when the beamtime is awarded, we arrange the logistics and the scheduling time, and we will send it to you through email. And you have to only send the doer and sample details and the method of experiment that you need for us to conduct, whether it is a native or MR, SAT, MAD, or CRRs. And also, if you have any heavy atom method that you would like to go for it, or even sulfur-phasing, all those things. And data will be collected and will be processed. And we will send you the instructions how to back up your data and the day after the experiment is done. And we will also follow up on further needs like structure solution, refinement, and validation and deposition during also the table submission and for the manuscript. So this service is provided at no cost, which is really great. And we do request including authorship for the manuscript and the deposition. And this program is only open to the non-proprietary experiments. And in this program, we have multiple projects work for this many years. And some of them are virus proteins like Zika virus, influenza, SARS-CoV related ones, and norovirus. And also we are working on a lot of targets, that is inhibitors and targets for attacking these virus. And also we helped people with the PKG protein kinase and chaperones and also computationally designed proteins. And some of the novice crystallographers who wanted to have the crystallization to be taken care and then the data collection, we do that as well. And so we also provide like SACS program along with the, like if you wanted to combine the SACS experiments along with the X-ray crystallography, we provide that too. And if you have any details, please contact us, and we have the email listed here. And thank you for the opportunity, and I hope you will come to us for collaborative crystallography. Thank you. Thanks, Jay, for introducing me.
Thank you, Banu. Yes, this might be actually very appealing to a number of users. One of the nice things about collaborative crystallography is you can fit them in between other beam times often. So it's a way, if it's hard to get time in, to get the data collected quickly. Okay, so next up, our next speaker is Greg Hura, who will be speaking to us via a video that he made for us.
Hi, everyone. My name is Greg. Wanted to start out with some basics for those of you who haven't done small angle X-ray scattering, maybe here for crystallography. Things I want you to know is that SACS is a solution-based technique, sort of the 30 microliter volumes, mg per mil concentrations. There are really no restrictions on buffer conditions, aside from melting the sample cell or something like that. So not a lot of sample prep. And then on top of that, it's high throughput, so you can collect a sample in a second. And so the layout of the experiments is very similar to crystallography. You will have a sample presented from the X-ray beam, solution, collect X-rays through it, and collect X-ray intensity as a function of scattering angle. And a typical profile shown there, area detector, but the data is circularly symmetric. So you can just integrate along a line and then just use the additional angles, circular angles, to get you a better signal to noise. So because it's a solution technique, SACS results are an average of all the conformations and assemblies in solution. So if you can get purity, you're ultimately going to get better resolution in either conformation or assembly. SACS provides about 40 unique pieces of information. Like, you can think about them as constraints, and they're through a wide scale in terms of distances. So everything from characterizing your 1,000 angstrom particle down to domains that move 5 angstroms apart. So sub-nanometer resolution. And it's been particularly empowered by recent structural modeling, structure prediction, homology modeling, and computationally driven conformational samples. And if you're looking for examples of its utilization, you can go to our website, civils.als.lbl.gov, which I'm going to repeat over and over again during this talk as the primary resource, and you can see great examples. So just to give you an idea of the scattering, the using the conformational sampling. Here you can see a protein undergoing conformational change in silico up there, PDB model. The SAX profile is calculated and compared against what was measured in solution. So you can pick out the conformation that was present in solution given this huge ensemble. And that's a very common use of small angle. So the two techniques we run, one is size exclusion coupled SAX. And to run size exclusion coupled, for instructions, you go to sybils.als.lb.gov. But you will book one shift. You will be able to run with one running buffer. And if you can have 7 samples in that running buffer, you can run that many per shift. And the typical volumes is 60 microliters. And what happens is you give the plate in from your EPIs, goes into a plate, the EPIs go into a plate. They get injected onto a size exclusion column. The flow through gets ported right in front of the X-ray beam, and small angle X-ray scattering is collected. And then you also get UV mols and refractive index there as well. So there's mass and things like that. So here's an example. Again, we collect areas. You can go by the intensity on that and get your typical elution peaks like you would get for UV. You can see this sample. There's a buffer region. There's 2 peaks here. If you average the results from this one peak, you get 1 scattering profile. This happens to be a heteromeric complex. So this is a monomeric version in excess, one of the monomers in excess. So that's that peak, and get the scattering curve from that. And then also you get this scattering curve from this peak. And if you zoom in on this peak, you can see that it's asymmetric and that the radius of gyration is changing across that peak. And so there are multiple species in here, and you can deconvolute that peak into the 2 species and get 2 SAX curves that describe 2 conformations or 2 assemblies that are hidden under here. So with 1 injection, you get 3 samples, 3 SAX curves, and can analyze them independently. We run high throughput SAX, and for that, we've coupled a liquid handling robot that picks samples from a 96 well plate that you prepare at your lab, and it pulls samples from each well and pops it right in front of the X-ray beam. It gets collected and then gets popped back into the sample cell, or into the plates. And the kind of things that gets done here is ligand screening, construct screening, condition screening. So here's, for example, a protein that undergoes a conformational change. Actually, it's a monomer to dimer transition upon NADH binding. So when you add NADH, it goes to a dimer. This is a titration. And then what this particular experiment was doing point mutations of various amino acids and seeing whether you could interfere with this transition based on the NADH. And some of them, were monomeric, and some of them were dimeric without NADH. So you can see amino acids that are involved in that transition. So last slide here. This is how mail-in SEC SAX works. This is at sybils.als.lbo.gov. We ask you to register twice, once for the rapid, which Andy will describe in a moment, and then once for the Sybils beamline. And you can do this thing for the rapid proposal once per year, just as long as your scope hasn't changed too much, and should be able to book slots within 2 weeks when the ALS is in operation. And that's, again, one of the benefits of having a remote data collection or mail-in data collection, is that we can push through samples a lot. Reminder that the ALS is typically down January and February, July and August. And we understand that there are many logins that you have to deal with. The rapid proposal is a different login from the Sybils proposal, etc. And then there's also one for results, and we're working through that. So we apologize for that inconvenience. But hopefully that explains the mail-in system. And thanks for your attention.
Next up, I'd like to introduce Corey Ralston. formerly at the ALS, but now on the bigger, better things with some cool, really cool new technology.
Thank you, Jay. Hi, everybody. I'm Corey Ralston. So I run the X-ray footprinting program here at the ALS. We're a very recent addition to this ALS-ENABLE resource. So we're somewhat of a new kid on the block here, but I'm really thrilled to be part of this awesome team. So I did want to start out with just a basic overview of X-ray footprinting because it's a lot of people have not heard of it and don't know what it is. So this is a very simple conceptual overview. So basically, it's a solution state method. We have protein in some kind of buffer. We irradiate with X-rays. The X-rays interact with the water and produce hydroxyl radicals. And the hydroxyl radicals will go on to modify protein side chains. They're highly reactive with protein side chains, and to some extent the backbone. So we get these covalent modifications formed on the proteins. And we then digest the protein, usually with a protease, and we then run our resulting peptides out through LC-MS to figure out exactly where those modifications occurred on the side chain. So this is the same overview, but in a pictorial form. So basically, we produce our hydroxyl radicals. They modify the protein in certain places where they're accessible. We digest, and then we run an LC-MS. And let me just tell you briefly how you actually get information out of this method. So imagine you've got a protein in two different states. I got one on the left side of the screen, one on the right side of the screen. And let's say you know the structure of your protein in the one on the left there, the compact form. And you run this method. And let's say you see a modification, which I've indicated by a pink circle there on the screen. And then you run the same method, the footprinting method, on the protein in its other state. And then you find that there's these modifications that occur on the linker between the two domains. And so from this accessibility information, from the hydroxyl radical modifications, you can infer that the two domains have come apart. And so that's how you get from solvent accessibility information to structural information. And we can do this on a residue by residue level. So you get information about the accessibility of each residue to water, basically. So just a couple highlights through the years. This method can be applied to a lot of different protein systems and nucleic acid systems. So it's been used to look at gating mechanisms in membrane proteins. It's been used by Sarah Woodson's group out at Johns Hopkins. It's done some absolutely beautiful studies on looking at ribosome assembly actually in vivo, so in cells. People have used it to look at GPCR activation. How does those proteins interact? People have also used it to look at antibody antigen binding, so figuring out where epitopes are. So it has a wide variety of uses. It mostly gives you interaction information, where are proteins interacting as a function of time. So practical aspects of the method. So we generally use micromolar concentration of protein. We usually need about 500 microliter volume to get a complete data set. We, like I mentioned, we're generally comparing the protein in one state to another, and this is because different residues have different reactivity to hydroxyl radicals. So you want to normalize that out by looking at the protein, say, alone versus binding to another protein or binding to a ligand or changing its conformation. One sort of drawback, I think, to the method is that we're quite buffer limited because pretty much everything you can imagine scavenges hydroxyl radicals. So glycerol is a no-no, detergents will scavenge radicals. Generally, we're doing our experiments in phosphate buffer, which does not scavenge radicals. So there's that limitation. I wanted to let you know the exposure times on the order of microseconds, generally these days on just 100 microseconds or so. So that gives you a sense of how much kind of time resolution we can get if you're interested in some kind of kinetic study. We do have to use LC-MS as part of this method, and that is at the moment a bit of a bottleneck for us. We have access to LC-MS and we will do that for users, so that's no problem. But we don't have our own instrument at the moment, and we have a very long queue, and we have quite a few user groups now. So this method is not fast compared to crystallography or SACs. We're very slow at the moment. So I just wanted to let people know that. We do have an additional inline fluorescence measurement that we can do if you're interested. So This is our setup at the beamline. We have a syringe pump to deliver sample past the X-ray beam, and we can use a jet, or we can have sample flow through a capillary. We have a lens for looking at your sample and also collecting fluorescence from the sample if you're interested in collecting that at the same time as collecting your footprinting data. So that was a whirlwind introduction to footprinting. I hope people are interested in the method, and please feel free to contact us. So I have a very, very small team of people here. You can contact me or Sayan Gupta. And you also see in that picture Lena Christensen, who's a really amazing research associate in our group, and Brandon Russell, who's our engineer in the group. And thank you. Thank you.
Yeah, once again, I'd like to thank Corey. And next up, I'd like to introduce Andy Jones from the Advanced Light Source Users Office, who will walk us through the process of becoming a user and setting up proposals for beamtime. Andy, it's all yours.
Thanks, Jay. Appreciate it. Hello, everybody. I am Andy Jones, also known as Andrea Jones. I'm at the ALS User Office as the Proposal and Publications Coordinator. I am your primary point of contact for submitting proposals and getting beamtime. In materials after this webinar, you'll receive a link to this page, Procedures for New ALS Structural Biology Users. This is an abbreviated form of our user guide, but it's kind of geared towards structural biology users to make things a little bit easier for you. Our user guide with all the information is on our ALS webpage. If you go to als@lbl.gov, you'll see this menu here for users. We get lots of information, including our user guide. So this is the full user guide where you can get all the details and more information if you don't have enough in that small guide. So the first thing you want to do to become a user is to create an account in our ALS Hub user portal. And we have a link here. And we have a link at the top of our user guide and a link at the top of our page. This is a screenshot of what it will look like when you go. I'm already logged in. So this is a screenshot of what it'll look like when you need to log in. So how you create an account is you log in with either ORCID or InCommon, and then give us your contact information, set up a profile. So everyone needs to authenticate their ORCID ID with us. So we suggest you log in with ORCID. It's the easiest way to do it. Some people prefer to log in with InCommon. InCommon is a service that we're about 1,000 or so institutions, including many research institutions and universities. You can use this to log in with your home institution's credentials if your institution is part of InCommon. You might choose to do that. You will have to authenticate your ORCID ID with us at least one time, though. So be prepared when you create an account to log into your ORCID account. Staff members with lbl.gov email addresses can use this. When you log in with ORCID or InCommon, you will encounter this page, this profile selection page. You must fill this out and submit it in order to complete your profile. If you're going to submit a proposal Sunday, you're not sure if you're going to submit a proposal, you can create your account today. Create it today, and then it'll be all set up for you if you're ever going to apply for beamtime. So when you get this question, what's your next interaction with the ALS? You can choose submit a proposal, which doesn't mean you're submitting one now. You can choose conduct remote experiments or create a profile. Any of these will give you a similar profile form that will set you up to become a user for remote experiments and to be able to submit proposals. You click next and you're given a profile form. You fill out your contact information. If you logged in with InCommon, you'll have to link to your ORCID account to get that authenticated. Give us some demographic and other information about your experiment. Click next at the bottom to review the form, and then click submit. And after you click submit, you should get an email confirmation right away that you've created a profile. And then within a couple days, our user processors will send you another confirmation when your account has been fully processed. Okay, the next thing you want to do is submit a proposal. I only talk about rapid proposals here, but I want to briefly talk about our two types of proposals. If you go to our user guide, apply for beamtime number 2 will give you information. Jay had mentioned there's general user proposals. These are good general user proposals. You can apply for beamtime on up to 3 beamlines. And you first apply for a 6-month cycle. It goes through review. And then after the review, you get beamtime that cycle or not. Usually, most structural biology users will. And then you can request beamtime for up to 3 more 6-month cycles. So this is a good proposal to use if you know you're going to be a user for a while. You can get beamtime on 3 beamlines for up to 2 years. So until we go dark, for our upgrade. So the next deadline is the first Wednesday in September, September 6th, to request beam time for 2024, the first half of 2024. Now, many of you are like, I don't want to wait till 2024 to get beam time, but you can submit a rapid proposal. Rapid proposals can be submitted anytime we have beam. Remember, we're shut down in the winter, January, February, and July, August. But when we have beam, you can submit a rapid proposal anytime. You can request time on one beamline for 1 6-month cycle. Greg Hura mentioned 1 year, but it's really a 1 6-month cycle. We just need some basic information about your experiment, a 1-page description of the scientific case. What's the scientific question you're asking? Give us some details about your experiments. Rapid reviewers like to know, do you have crystals on hand? Are you ready to go? The review process takes about 1 to 2 weeks. It's fairly quick. And then if you're allocated beam time, or you can contact the beamlines and discuss scheduling of your proposal. For general user proposals and rapid proposals, if you're not sure which beamline you want to apply to, select beamline ALS-ENABLE, and the ALS-ENABLE beamline scientists here will assign your proposal to the appropriate beamline. You can also request collaborative crystallography that Banu talked about. Okay, so your allocated beam time, we will check and make sure that we have a user agreement with your institution. Now, safety documents. Every experiment needs an experiment safety assessment form. Some of the ALS-enabled beamlines are nice and will submit safety forms for you. But sometimes you're asked to submit your own safety forms describing your materials, your samples for the safety review. The ALS user office prefers that users do this themselves, but some of the beamlines prefer to do this. So you may be asked to submit an experiment safety assessment form. If so, you will receive an email with instructions on how to do so. You get there from ALS Hub. When you're log into ALS Hub, all the links to submit proposals and complete experiment safety documents and things are all here. Now, most structural biology users conduct their experiments remotely. Mark Allaire showed you some great software to conduct your experiments remotely, but some users do come on site. If you come on site, you do have to register an ALS Hub. You have to log into your ALS Hub account and tell us when you're coming so we can get you through the vetting and badging process. If you come on site, be sure to go to our user guide, and there's a link there in that 1-page document. Here, to number 5 in our user guide, get access to work on site. If you're a US citizen, you need to tell us at least 3 weeks before you come. And if you're a non-US citizen, we need 6 weeks notice before you come. How you tell us you're coming is you log into ALS Hub, and where it says arrival timeline, you click update here to tell us when you're coming, or you click update your profile to tell us when you're coming on site, or you click profile and say you're going to conduct on-site experiments. All of those ways will bring you to the same form where you give us your arrival date and estimated time of arrival and some other information that we need to know to process you to get your badge. You might want to come a business day or so before your experiments to get badged, just so when your experiment starts, the next day you're ready to go. Talk with the beamline scientist if that's appropriate for you or not. If you do come on site, you'll need to do some safety training. This tells you about all the safety training you need. You go to ALS Hub, to the homepage, to actually do your safety training, complete online safety training. There's also other resources available to you. The lab has a guest house, a hotel where you can stay. There's also food resources, lactation rooms, and all those things. You can find this out at available resources. After your beam time, we'll email you to complete a user satisfaction survey, whether it was remote or on site. We also ask that you report all your publications to us and acknowledge the ALS. And so if you click here and go to our publication menu, the appropriate acknowledgement statement for ALS-ENABLE to include in your publication is right here on the website. To report publications, just click here, put in the DOI, hit continue, put the beamlines that you used, your funding sources, and you are all set to go. And I think that is it. Once again, you will get a copy of this after this webinar, and you can always find our user guide on the ALS webpage. And under users, if you go to user services office, I am Andy Jones, Andrea Jones, the proposal coordinator. You can find my contact information here to ask me questions anytime.
Okay, I just want to finish up by saying just a couple words on outreach and training. I'm the PF for the outreach and training part. We will train every user that needs to use their beamlines. That's a given. And I think our crew really enjoys doing that too. We have very good teachers working with us. We could do this on-site or remotely. We're also putting together a number of documents that you can access via the web. We like to make sure that we're including as many graduates and undergraduates as we can. One of the ways we do that is through having people link us into their seminars or even courses so that we can become a part of what they're doing that day. We do that a lot with high school visits, undergraduate visits, but we've also incorporated a number of our beamlines into biochemistry courses and capstone biochemistry courses at Caltech, University of Missouri, so that the students actually get a chance to, collect their own data as part of an undergraduate course. You'll find us at about every ACA meeting. Look for our booth. Probably have a presence at the IUCR this year also. We do local workshops. We have, like I said, virtual training. We also offer virtual office hours. So if you have a question, just generally something coming up, something you're working on, or some data you've already collected, let us know and we'll set up an office hour so that we could just go ahead and talk about that. And that doesn't matter whether you collected the data here or not. This is just a general thing. Of course, most of the time people are asking about specific beamlines that we run ourselves. We're also working on lots of coordination with the other light sources, especially with our colleagues that are turning off the lights next week. They are still a great resource for contact and trying to get beam time because they know us. They'll send an email, they'll give us a call. I'd like to plug our ALS-enabled webpage, and that's at als-enable.lbl.gov. You'll find links to all of our beamlines. You can look at per beamline. You can look at us via what technique you'd like to use. We've also got news and science highlights, training materials, and links to individual web pages, which is really great if you want to get to the civil stuff, because civil's the BioSACS group, they just have tremendous resources available through their webpage. So just as a summary, we all do standard on-site MX at our MX beamlines. We do also offer room temperature and experiments and plate screening. BCSB has automated data collection. So basically you can think of us as one meta beamline when you think about your proposal, right? If you just choose ALS-enable, like Andy was saying, we will triage this. We, as the beamline scientists, look at each other and decide where the best place for it would be. Collaborative crystallography, if you contact Banu or myself, we'd be happy to walk you through that process. Greg mentioned his small angle scattering work, and you could contact either him or Mikkel Hamill from the Civils group. Corey, X-ray footprinting, extremely exciting stuff. It's just really fun watching that program take off here on the floor. And you can contact Corey as she expands this program. Just another note, if anybody's interested in becoming PRTs, the thing about PRTs is, like I said, it gives you the most flexibility in getting beam time. Most of us call it on-demand beam time for our PRT members. I think the fastest I was able to get somebody up at 422, they contact me and they had their crystals inside the beam within 7 minutes. And I felt bad because I promised them 5, but I got caught up in the hallway on my way downstairs. And it's extremely flexible. Industrial users, the PRTs have a number of ways for you to gain access also. But basically, if you contact us, we could walk you through that process. So gaining access for both general user proposals and rapid proposals, make sure that you register as an ALS user. Also ensure that your institution has an ALS agreement. So one of the things we do look for is what institution you are on, what agreements we have in place. As far as rapid proposals go, most of us have increased the percentage of rapid proposals we're allotting during the APS dark periods, since so many people are going to be jumping in for the first time. Yeah, with that, I'd like to thank all my speakers. I'd like to thank Paul for getting this all put together. And I'd especially like to thank Catherine Burnett and Corey Ralston for help putting this whole thing together and ensuring that it runs smoothly. With that, we've left some time for questions and answers. I believe we are going to be doing this via the chat window. Is that correct, Kath? Yes, so if you have any questions, please feel free to put them into the chat and we will read them off and answer them.
Chat or Q&A?
Is there a difference? Oh, there's a Q&A section. I'm sorry. There's a Q&A.
So you want to answer one of these live? Does that mean that you?
So we have a couple of questions. Maybe we should just go through them.
Right.
Sure.
So someone was asking about, I guess that's been answered, 18mm pin questions for answered. Okay.
I just answered the HHMI question.
You want to just repeat out
Yeah,
loud?
would you like to put these out loud?
Yeah, so in order to get access to the HHMI beamline, HHMI is providing us a list of investigators that are allowed to, and then we could just check your name on the list and get back to you in that regard. So.
And I'll mention this about PRTs, right? You may be a part of a PRT and not even know it. This happens to my PRT all the time. So I often get people from the University of Iowa especially, it's like, oh, we're a part of this? Talk to people, talk to a beamline scientist, let us know where you're from, and we can tell you if you're already a part of a PRT, if you have some kind of PRT access. Shadon, hey Shadon, how's it going? It's been a while. So he's asking James about metal scanning. Can you tell if the metals are in the crystals or the solutions?
You can, well, he said and, so, but the long and the short of it is you can tell if the metal is in the beam.
Yes.
So if there's metal under the beam, it will light up in fluorescence. The way to tell if it's found in the crystal, that's a different experiment. You want to collect diffraction data. By the do the metal scanning thing, the crystal can be dead. It doesn't have to diffract at all. You want to collect diffraction data above and below the edge of interest. And then it will either light with an anomalous signal in one and not with the other. There's nothing else in the periodic table that will do that. So it's actually one of the best ways to definitively identify a metal in a sample.
A lot of people back. So I also recommend if you're collecting and you're processing data and you're just doing one ounce of native data and you've processed and you get the message that says, hey, we saw an anomalous signal. You probably did. This is a quick experiment you could do after processing. Just throw it back up with the robot, do an elemental scan, and it'll give you an idea of what to include because, I mean, it's experimental phasing information, and that's always worth including.
The question is going to be hard to answer. Based on the number of proposals you've received, will you see decreased availability at ALS-ENABLE after the APS shuts down? That's a good question.
We're in clear.
Yeah, we don't know. We can't. I guess we can't answer that.
You know, well, Catherine, you're closer to the proposals than we are. But.
Yeah, I feel like we have a lot of time, especially I think both 831. The best thing to do is come and we'll see. We'll do our best to fit you in, definitely.
I mean, I can say the mathematics of it, 90,000 hours of beam time are going to go away. So yes, we'll have a lot of people wanting to collect, but we haven't seen that yet. So it's not too late.
Yeah, and we're also not just talking about our individual beamlines and our individual programs. With ALS-ENABLE, we are really working together and sharing resources, including staffing, so that we could get more things up at night, turn things around better. If I'm slammed, you can ask Mark or James, hey, this experiment needs to go. Do you have time? That kind of thing.
If you are a member of CERCAT, then CERCAT has secure beam time on BCSB beamline, so you could just contact us or contact your CERCAT manager, basically.
He might be a member of PRT's PRT, not even know it. The anonymous attendee.
Oh, I think,
That one from, well, that was a conversation about the detergents in.
and I suspect that's an answer from Cyan, who's being very precise about how much detergent you could put in your solution for footprinting. But like he said, it depends a lot on the water content of the protein that you're trying to get structural information on. So I recommend you talk to us, email us, and we can set up a Zoom to talk with you and learn more about your system and try to figure out if the footprinting is worth trying.
Okay,
A
so,
question from Bayer.
Yeah.
yeah,
Mark, you want to do that?
yeah, Mark, no,
So
Mark.
which one, which question?
The work at Bayer,
Timothy Rydell.
Timothy Rydell.
Okay, let me read that and I'll get back to you. You could answer a different question as well, but I'll read that one.
Collecting data from long unit cell axes. Is there any special setup for this? You want a large detector,
Yeah.
or you want to turn up the energy, or maybe a combination of both.
And you can, you could use also to shift the detector up if you're pushing the detector far back in order to achieve the resolution. And there's a few beamlines that does that at the ALS.
Detector angle.
Yeah, that's right. The vertical one shift.
We have Q315s, so.
No, they're all off before.
So unfortunately, the Polaris detectors have fewer pixels than the CCDs did. So that does make this a bit more complicated, but large unit cells are not that much of a problem. Large unit cells that diffract really well, that puts a challenge for detectors, but it's still something that, again, when you put in a proposal, describe the science you want to do, and we'll help you with that. Not exactly how big is the unit cell, how well it's diffracting.
Right. We also have a number of beamlines that have kappa or mini kappa capability if your spots like to line up in a specific orientation. That's just terrible for data collection. You know, they're all standing up and they want to rotate in. Getting that off axis can help. But yeah, that's another one of those things where tell us what you got and we'll talk about where the best place to put it would be. Mark, do you have something?
I'm sorry, no.
Oh,
Read the question.
you don't want to say that now?
Yeah, I think there's a question about data process, CIRCAT, and data processing and accessing BCSB beamline. I'm going to answer in general that any data sets that I think all the ALS-ENABLE beamlines are automatically processed when the data set is collected. This thing takes about a few minutes, and then at the end of the day, you have your data reduced and MTZ file generated. So.
I'd like to just make a general comment on automated data processing. I love it. It's great. It usually does a very good job for standard crystals, and you should never stop there. It should never be the automated data processing results that you're submitting for your PDB or to solve your structure. Go back, look at the logs, you know, give it one final round of reprocessing, you know, get that ISA up higher, you know, make that CC 1/2 better. You can always get a little bit more. And sometimes, you know, it will really help where, you know, you gain a half angstrom or something like that, or you find out, oh, it's the wrong space group unit. So every time you get, and this is general, anywhere you get an automated data processing result, go through it again.
So the second part of the question is about allocation and the number of hours per shift that is being done, especially for industrial and proprietary users. So contrary to the APS, the ALS is working with the minimal shift of 4 hours from a proprietary point of view. So I think if you run less than 4 hours, this is going to be the minimal charge that the ALS will do. So we go by half shift to some extent, so half shift, 8 hours, and so on and so on.
So Timothy had a follow-up question about the auto processing, asking if you can people log into computers and reprocess after their beam time?
This is allowed to do as long as you have the appropriate license for all the software that you're going to use. So all the beamlines are basically using SBGrid to provide all the software that are available through the SBGrid umbrella, including XDS and dials, basically. Let me correct, so XDS is coming directly from XDS, but the dials package is coming through SBGrid. And all the data can be reprocessed, but again, from a proprietary standpoint, be sure that you have the appropriate license to do that. So.
I think Andy answered another question. Yeah, great. Did a good job triaging all these questions.
So we do have a couple of minutes left in case anybody would like to get a final question in. Does anybody at the table have any final thoughts before we take off?
We will just remind people, we'll put this recording on the website at some point.
Oh, can we use HKL 2000 for data processing? That's a general question. I would recommend talking to each beamline scientist. We each have a slightly different set of suites. 422, we use XDS and CCP4 suite programs.
So also I would have with default settings, HKL does not handle highly dose slice data very well. It can if you contact the author and ask them the magic keywords for doing it.
And that was one of the things from 422 when Shuttle-S, that XDS was turned out to be the processing software of choice by my consortium members.
So I can add that for the BCSB detector right now, or attempting to get the dev site file for all the detectors that we have, so that at least you could get a first start in using HKL if you want to do so.
Yeah, that's a good point. Most of us do have dev.site files that we can give you for our detectors. If you have HKL 2000 and would like to process it yourself at home.
Good question from Jeremy.
Are subscription ratios for the various beamlines available somewhere? Ratio of applications is shift supported? I don't believe so.
Not that I know of.
One of the things that we do, even if you do not choose ALS-ENABLE as a beamline during these general user proposal allocations, is that what we will talk is like, oh, this proposal didn't get anything. One of us will chime in with, well, I've got a little time, why don't you give them a shift over here at my beamline, kind of thing. So just give it a shot and give it a best shot, whether you want ALS-ENABLE to triage where it should go, or just take a look at the beamline specs and put it where you think it should go. Contact the beamline scientist. And we really are in the habit of saying yes and getting people as much beam time as possible. A lot of things don't score well because they're so early in the thing, but that's some of the most important beam time you could get is at the beginning of a project to help, get everything optimized in a reasonable amount of time so that you can get the project done.
I could add that at this point of time, the subscription ratio would be a little bit biased because a lot of those beamlines have been upgraded with detectors and now they are performing to the level of all the other beamlines. So I think that's the good thing at this point. We have all our beamlines upgraded so that we can support the wave of new users that will come from the APS particularly. So.
I think right now, as it stands, if you want the midnight to 8 a.m. shift on Saturday morning, you can get it.
Unless I'm collecting. So there's 422 won't be available because that's my favorite time to collect.
So this is another point that James and Jay are just talking, is that indeed at the ALS we have support really from sometimes 5 a.m. in the morning down to midnight in the evening. So we're making sure that at least you will get support when needed. And the most critical thing is that we don't like to lose any crystals from the robotic system. So we're making sure that all of our beamlines are well equipped so that if anything is going wrong with liquid nitrogen, the due word, then we can act rapidly to be able to come and save the samples. So we know how precious are those samples to you guys. So.
Okay, well, should we call it? We're down to only about 50 participants now.
I can never see, what, I don't know how many numbers were there,
Oh,
but 50, I didn't expect that
we
many.
have 104
We have
from
108.
108.
Still, I'm looking half glass full. Thank you, everybody. Interesting times are upon us in the synchrotron world. We all band together and help each other out. I'm sure we'll all make it through okay. ALS-ENABLE is here for you. We like to say yes to programs. So, you know, don't hesitate to contact a beamline scientist. We'll get you pointed in the right direction.
Excellent. All right.
Thank you.
Thanks, everybody.
Thank you, everybody.
Thank you.